Excellent. Thank you, Shannon. And it's great to have this conversation with Charles Raison. I'm guessing you go by Chuck. I mean,
I
based
do.
on
I do.
so Chuck. We generally like to chat a little bit about you and how you arrived in Wisconsin, I guess from the Southwest, to just sort of begin our conversation. So maybe just give us a little bit of a trajectory of your arriving to Madison, and we can start there.
Sure. Well, right. So I'm a psychiatrist that was a clinician starting out in life, but have been mostly a researcher since the turn of the millennium. And over time, increasingly working on these things I sometimes call ancient practices. So trying to see whether ways that humans sort of worked at enhancing well-being across historical time might be retrofitted for depression. And I had done some early work with meditation and heat hypothermia. And I was in Arizona, and through a series of just people I knew, in 2015, I got a call from a gentleman named Bill Linton, who had established a nonprofit medical research organization. It was also really a drug company, basically called USONA Institute, looking for a psychiatrist to head up their nascent program on pursuing psilocybin, which of course is the psychedelic you see in quote magic mushrooms, pursuing an FDA development program for psilocybin. And so that's how I came to Wisconsin. I wept some tears around the weather. I'm from the West. I'm from California. It was hard to leave the West. I've had to do it several times in my life. But anyway, so I came to Wisconsin, and I had this sort of interesting position. I'm half time at the University of Wisconsin, where I'm a professor, and then I have a number of other positions that all sort of interact with each other around themes related to psychedelics and depression and a little bit of spirituality. But one of those is that I'm the director of clinical and translational research at USONA. So much of my life these days is in conducting this sort of palette of either more translational basic mechanism or human mechanism type studies, trying to understand how psychedelics work, and then drug development stuff, helping contribute to these large phase 3 type studies. And I still do a little bit of research. I'm the director of a new research institute out in Vail, Colorado, and we're doing an interesting study of heat out there as an antidepressant modality. So anyway, quick, short answer of how it's much more, there's many more twists and turns. That's basically how I came
They're
to
very, very cool.
be in Wisconsin.
Maybe you could give us a little bit. You mentioned depression and you mentioned cancer, and I know that you've done a good amount of work on inflammation. Can you maybe just give us a little bit of background on the relationship, because it may be very new to many, on the relationship between depression and inflammation, and possibly sort of the evolutionary context of that?
Yes, absolutely. And of course, MD Anderson has been such a centerpiece of this. It's really interesting. I did that work at Emory University from about 1999 to 2011 with Andrew Miller, who is really one of the leading world stars in the space. And Emory was one of the places, along with especially when Robert Dancer came to MD Anderson, one of the places where we began to realize that these processes that we thought of as being bodily processes and really thought of initially as problems, inflammation, that these evolved ways of fighting infection, while they certainly contribute to humans surviving infectious agents over evolutionary time, also had a real proclivity for inducing depression. So we began to, originally back in the 80s when psychoneuroimmunology was born, the early studies suggested that depression was a state of immunosuppression. And so it was really an iconoclastic move, and Robert Dancer was really one of the people at the start of this, where there was this realization of, whoa, wait a minute. On other measures, people with depression as a group seem to have increases in immune activity, in particular, increased circulating pro-inflammatory cytokines. And so there's a whole huge literature showing that if you take groups of depressed people that are otherwise healthy and compare them to sort of matched people who are not depressed, the depressed people as a group show these modest chronic increases in inflammation. Around the same time, and Andy really was one of the people, head gates of this also, there began to be this realization that if you gave people who were not depressed agents that activated inflammation, like say interferon alpha, many people got very depressed, got depressed very quickly. And so that was the sort of evidence that, whoa, you can take people, if you inflame them, they tend to become depressed, develop depressive symptoms. And then over time, we had the idea that, well, if that's the case, maybe powerful anti-inflammatory agents would work as antidepressants. So we did a study where we gave people who were healthy but were depressed. We gave them a drug called infliximab, which has very specific, very powerful anti-inflammatory effects, and really nothing else, and showed that it did not work as an antidepressant, interestingly enough, in the group as a whole, but that there, in people that were depressed and had increased inflammation, it did work better than placebo. And at that point, I kind of peeled off and thought I was leaving the world of inflammation, which turned out not to be true. We've got some very interesting findings on psilocybin inflammation. But the Emory group went on to show that, in fact, it's not that depression is an inflammatory condition. You can be unbelievably depressed and have very low levels of inflammation, but that inflammation is a pathway into depression, and that there is a subgroup of depressed people wandering around who have chronically elevated inflammation, and not like you get with the flu, but modestly elevated inflammation. And those people have different, their brains are working differently. They respond differently to medications. And so I think really it's the best subgroup we have yet in mental health for saying, hey, this is a group of people who looks like, they look like they're depressed, but they actually have a slightly different underlying biology. And so that's really kind of where the field has mostly sat. Now, of course, this is directly relevant to cancer and its treatment because both tend to induce inflammation. And so that is a subgroup of people or a population of people for which this connection between inflammation and depression is probably especially germane.
So, it's super interesting that this idea of inflammation and depression, um, inflammation is such a broad term, right? And
Yeah.
you can have sterile inflammation, where it's not infection associated. You can have acute inflammation from a microbial infection. I just wonder, with these sort of this correlation between inflammation and depression, are there signatures? And you mentioned these that there are some different types of subgroups. Are there signatures of inflammation that can help you sort of sort out these types of trajectories, I suppose?
That is a really interesting question. I think the short answer is no. Um, I mean, there have been papers over the years claiming this or that, but no, I mean, I'm interested in interleukin 6, right? Because it's this interesting cytokine that floats around at higher levels in the body, so it's easier to measure. And it's very complex, right? So generally, IL-6 is really, really a bad guy. So it's been probably the marker that's been most identified with depression, maybe TNF or IL-1 beta after that. But no, when you look at these, these things tend to travel in packs, right? So, generally when you get something that activates an inflammatory process, you tend to see a lot of these things tracking together. And then, of course, these pro-inflammatory cytokines activate anti-inflammatory cytokines.
Mm-hmm.
So they also often track together. When I said that I had wandered out of the world of inflammation, one of the places I wandered, as we were sort of talking about, is into this idea that hyperthermia, heat, which of course has been used in cancer, especially in Europe, right? That it
Mm-hmm.
may have antidepressant properties. And we and others have now shown that that does seem to kind of be the case, that if I put you in one of my special little heating devices and cook you half to death, you're likely, if you're depressed, to feel a lot better for a couple of weeks. Well,
Mm-hmm.
we measured cytokine responses, inflammatory responses in the blood to that treatment and found that it didn't really activate anything except IL-6. Now, this is interesting, right? Because that looks a lot like exercise, which also sort of seems to differentially activate IL-6. And it's surprising that an anti-inflammatory strategy would kick up this sort of, quote, nasty pro-inflammatory cytokine. But in the study where we best characterized it, it's not a large study, it needs to be replicated, but the more the heat kicked up IL-6, the more undepressed people got. And of course,
Mm-hmm.
IL-6 signals in ways that are both pro-inflammatory and anti-inflammatory. So again, just pointing to this fact that it's really not so clear. And so if you look at, say, the work that's coming out of Emory, which is the work I know best, it doesn't, you can see the signal with TNF or CRP or IL-6. There doesn't seem to be like depressogenic cytokine. And so this is very interesting. And then again, something like IL-6, which really tracks nicely with depression, it tracks a lot with the sort of changes in the brain you see in these sort of folks that have increased inflammation and depression. In our hyperthermia work, we seem to be able to isolate it from some of the bad guys it travels with. And there it seems to have perhaps very different effects. I don't know.
Mhm.
There's a lot yet we don't really
Well, I mean,
understand.
I think, it sort of speaks to this idea that inflammation always gets a bad rap. You mentioned IL-6. IL-6, obviously a myokine from muscle,
That's right.
is pretty essential for muscle regeneration early on. But if it sticks around too long, then it becomes sort of
That's right.
a
That's right.
problem kid that doesn't leave the party.
That's right.
And
And there are studies showing that if you block that inflammatory response, just generally with like an NSAID, you don't get the muscle growth. It really is
mhm.
interesting.
So timing and homeostasis. So this whole idea of psilocybin. I mean, we have to talk about this. It just sounds too interesting. It reminds me of Michael Pollan and his book on, I think,
How to change your
um,
mind.
yes, 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 and how in the context of life, like end of life scenarios and severe cancers, that there's often depression, and some psychedelics like psilocybin can play a pretty important role in helping mediate sort of a spiritual and psychobiological uh, sort of mindfulness, if you will.
Hmm.
Could you sort of speak to the psilocybin and in the context of cancer therapeutics and
Yo,
what you guys are up to?
yeah, I can speak about that all day, so just shut me up. Um, right. So, and there's just an article out by Michael Pollan today in the New Yorker about spirituality
I remember
and
that.
psilocybin. It's really interesting. But psychedelics had this sort of first flowering, for good or bad, in the 50s and 60s. That the research goes off the rails, gets into the culture. Nixon closes it all down. It kind of dies as a research enterprise, floats around in the underground a little bit. But and then there's this thing called a renaissance. Maybe that's overly grandiose, but research picks up again in about 2000 when a friend of mine named Bob Jesse brings together people and convinces a guy named Roland Griffiths, who sadly passed away from cancer in the last year, who was a well-known psychopharmacology researcher but who had an interest in meditation, to begin to look at psilocybin. And so he starts very carefully giving psilocybin to healthy volunteers and showing that it seems to have these long-term psychological benefits. And so, somewhere around 2009, maybe 2010, somewhere in there, he gets together. He and some and folks at NYU say, well, what's the next thing we can do? We think this may have therapeutic potential. It's so stigmatized. Where can we go? Where can we go where people, it's sort of like that old, ad about the cereal. Let Mikey try it. Like, who can we give it to? They're going to die anyway, and there'll be a lot of sympathy for them. How about people with life-threatening cancer? So there were these two small studies that were started around that time, around 2010, I think, one at Hopkins, one at NYU, where they took folks that had a potentially lethal cancer and gave half of them a placebo and half of them a high dose of psilocybin, and then measured their depression, their anxiety, their spirituality, all this stuff. And showed in two papers that were published in the same Journal of Psychopharmacology in 2016, that compared to the placebo, people that got this single high dose of psilocybin had these profound experiences, many of which seemed spiritual by any understanding of that word, but mostly that their depression and anxiety dropped considerably, and they had this sort of crossover design so that eventually all the people in both studies got a high dose of psilocybin. And when they followed them up 6 months later, like 70-80% of them were in remission from their symptoms, and especially in the Hopkins study, these people were depressed. They could have gotten into an antidepressant trial, like an FDA antidepressant trial. So this looked like this crazy miracle, right? Like one
Mhm.
dose of the medicine that's in your body for, I don't know, 8 hours, and 6 months later, you're quote cured. And many of these people said, yeah, we were devastated by the fact that we were dying of cancer, and we had this incredible psilocybin experience during the quote trip, where it caused us to rethink our relationship to death, to cancer, to mortality, and it sort of took away the problem when it did that in so many of these people. So really, the this all the psychedelic stuff that's happened since then, this explosion of funding, interest, and commercial development, and all this really was born of these two smallish randomized trials in cancer. And so cancer and psilocybin and cancer and psychedelics have been bedfellows from really the very beginning. And now NIH has taken an interest in this and has funded one study, and I think, knock on wood, is about to fund another one, looking at just this question. And of course,
Mm.
Moran and the folks at MD Anderson are doing these remarkable studies in people not necessarily that are dying of cancer. You can talk more about it, but in the context of cancer and showing really, really intriguing results. So honestly, and I've said this in multiple venues now for several years, if I were to make a list of conditions or situations that I think psychedelics can be most beneficial for, even though I study depression, I don't think that's going to be the most significant condition that's going to be of benefit. I think end of life. If I had to bet on one thing, if like I could take all the resources,
Mhm.
and say, let's chase one thing first, I would chase end of life. And not just necessarily in cancer. There's some intriguing data from Australia that is just out suggesting that no matter what you're kind of dying of, these agents, they give people experiences that really seem to provide them with comfort.
Yeah, I
But yeah,
mean, if I could just, sort of follow up on something. So, in your prior work, you've been doing a lot of work on inflammation and the relationship between inflammation and brain states of depression, and now moving into psilocybin. I just wondered two things. In those early studies and maybe the ongoing studies, does psilocybin have an effect on the inflammasome or inflammation levels?
yeah,
One, and two, does psilocybin in any way cause neural reprogramming, so that that there's a transition in the way you're processing neural activity? Just if you could just sort of
yeah,
speak to the circulating inflammation and maybe neural
yes,
effects.
yes, I can speak to that. And the answer is who knows, as in so many things. But I can tell you kind of what. So, and this is my gloss on the overall literature. If you say we're going to give people psilocybin and measure their blood before and after and look at circulating protein biomarkers, do you see some sort of major shift? I think on balance the answer is no. I mean, some studies see nothing, some studies see an increase, some studies see a decrease. Uh, don't know. Um, if you look at animal studies, I think there's some very intriguing data from several groups that psychedelics may have an anti-inflammatory effect on the brain, that they may truncate sort of microglial signaling and sort of astrocytic activity that promotes central nervous system inflammation. So I think there's something there.
Mhm.
We have not published it, but I will tell you that we have been looking at gene expression analyses with Steve Cole, who's done a lot of
Sure.
this work at UCLA, right?
Transcriptional signatures of stress. Yeah.
Bingo, right. And we find so it's really interesting. It's a study of 104 people that were randomized to one dose of psilocybin, a high dose, or placebo. We see a very strong, immediate, and long-lasting reduction in type 1 interferon signaling that correlates with reduction in depression. It's one study, haven't published it. It's a secondary analysis, but it's very intriguing and it sort of makes sense because, as we were talking about a second ago, type 1 interferons are probably the most depressogenic molecules known to humans, right? And so there's potentially an interesting story there. It needs to be replicated. So I do think there's something there, and I, because of the world I hang out in, I can tell you that I know of some colleagues that have done work in sort of longevity models in animals, and there seems to be this emerging signal that psychedelics may have life in sort of longevity promoting type effects that are really quite striking in the animal
What
model.
researchers in the longevity space are you referring to?
This is it's
Oh,
not
it's
out there yet.
secret. It's top secret.
Can't say it, but keep
Oh
your
well,
eyes on
at the Cancer Neuroscience Symposium, we'll offer you a
it.
drink, and hopefully we'll.
Well, and by then I can probably wave the papers in front of you. Sadly I'm not on any of them, but
So maybe Chuck, you could speak more to the neural plasticity that
yep,
may be affected by psilocybin.
right. So this is the $1 billion question, right? You think about what does a psychedelic do? You take it, you have this high dose, right? You have this wild psychedelic experience that people often find incredibly meaningful, even though it's often very strange. And then in studies, almost 100% of the time, you see a drop in problematic behavioral patterns, be they substance abuse or PTSD or anxiety, or in our work, depression. You see it within a day, and it tends to last, for weeks or months, sometimes depending. So what's going on? It's got to be doing something in the brain, right? So there are these two sort of different schools of thought on this, and this is, if you ask me, like, what am I most obsessed of in all of life, science-wise, this is it. So you get these conscious experiences that people find very meaningful, and if you ask people to rate their conscious experience along certain axes like mystical or emotional catharsis, if you ask them to score their experience the day afterwards, the higher they score it on those metrics, the more undepressed they are, say 6 weeks later or even 6 months later. And that sort of suggests, wow, maybe it's like psychotherapy. There's this sort of cliche, like a year of psychotherapy in a day. Maybe there's something about that conscious experience that's causing a long-term change. And if you say, well, how is that possible? Roland Griffiths himself used to say this, sort of like anti-PTSD, post-traumatic stress disorder. You can't, to get that, something horrible happens, you're conscious of it, and then you're different after that. We think your brain's different. But on the other hand, in animal studies, if you give rodents, or monkeys, as it turns out, some of the work being done at UW-Madison, if you give a rodent, though, high dose of psychedelic, its brain lights up like a Christmas tree, and you see this explosion of synaptogenesis and neurite growth. You basically you get this. This is the neuroplasticity, right? And you see it in brain areas that you'd want to see it in. And so, there is a reductionist camp, a non-conscious reductionist camp that says, it's the hard problem of consciousness. How could something immaterial like consciousness, impact a closed system like physicality? Therefore, the consciousness is sort of epiphenomenal. Probably, it's not really doing the deed. That it clearly must be that these agents are powerful neuroplastic type or neurogenic type agents, and this has led now to one of the primary kind of things going on in the psychedelic field are folks that are trying to develop what have been called neuroplastogens or psychoplastogens, which are agents that they started with a legacy psychedelic and kept tweaking the molecule and giving it to animals to see, can we find something that has massive neuroplasticity but without any of the psychedelic stuff? And if we did that, would it work as an antidepressant or whatever you want to use it for? And then we can do away with the psychedelic experience, take it at home, and so there's a huge interest in that, precisely because of this sort of finding in animals, these neuroplastic effects. Much, much harder to find that in humans. We don't cut people's brains open generally. And the data on what psychedelics do long term to the brain is really just not there. It's obscure. I mean, people have tried to look at it, and again, the findings, there are some findings, but they contradict each other. They're not replicated. It's been very hard to do that. So this is, in my research at W-Madison, this is what we do. We have studies where we try to separate out the consciousness from the underlying, non-conscious biology to see if we can understand how much of it is just neuroplasticity versus, it could well be that the states of the brain that induce the neuroplasticity also have to induce the consciousnness. You can, so we don't know. So again, in animals, lots of evidence. In humans,
Yeah, I mean, it's just super fascinating, and you mentioned how there may be different uh, types of people that could be responders or non-responders to psilocybin type of intervention, and it makes me think of like pharmacogenetics and pharmacodynamics, and whether or not the receptors for the active version of psilocybin,
psilocin, yeah.
whether there's polymorphism in the population that could influence the responsive phenotypes
Yeah.
to psilocybin treatment. And I wonder if there's any thoughts on that type of
Yeah,
research.
there are. There's another debate, which is, well, how do these things work just molecularly?
Yeah.
And the standard brand answer is that they are agonists at the 5-HT serotonin 5-HT2A receptor. It's a postsynaptic receptor, and that is based on the fact that there are antagonists of that receptor. And if you administer an antagonist of that receptor, you can block the psychedelic experience. There's one study I know, one PET scanning study from Scandinavia, uh, where they looked at 5-HT2A receptor occupancy in human brains after they gave people a high dose of psilocybin, moderately high dose of psilocybin, and found that it does, of course, indeed occupy the 5-HT receptor. And the more it did that, the stronger the psychedelic experience. Given that the psychedelic experience almost always associates with longer term outcomes, it does suggest that receptor occupancy, that there probably is some relationship between your sort of activity of the 5-HT2A receptor in the brain and longer term therapeutic effects. Um, where there are polymorphisms for the 5-HT2A receptor, people are looking at that. I think that this is sort of one of those open questions, whether or not you need to dose people higher or lower, depending on that. I have not seen in the literature. That doesn't mean, even now in this literature is so large, I don't see everything. But I have not seen anything that really supports that. Unfortunately, in psychiatry, pharmacogenetic things have not really yielded or not delivered on their promise. And so I'm always a little bit, what's the word, suspicious or cautious about, the sort of idea that they're going to do that here. But it's interesting now, and these agents do follow a dose-response relationship, though, as far as we know. So there are two large studies that had some sort of dose-response design. Compass Pathfinder did a large study of high-dose psilocybin in people with treatment-resistant depression. They had a 25 milligram dose, a 10 milligram dose, and a 1 milligram dose, and they separated nicely. So the 10, in terms of antidepressant effect, landed right between the 1 and the 25. Then my buddy Rob Barrow, who is the CEO of MindMed, designed a very nice 5-arm study looking at placebo versus escalating doses of LSD for generalized anxiety disorder, and showed also an interesting but nice dose-response relationship. So there's definitely, up to a point, definitely the higher the dose, the better the therapeutic outcome, and by implication, probably the more you're stimulating those 5-HT2A receptors.
Well, I'm curious about that signaling and whether or not the signaling through these receptors and whether those receptor levels differ in different states of cancer disease or depression. For example, would you mentioning end of life? I mean, are there any data suggesting that those receptor levels and therefore susceptibility to signaling through those receptors may change with disease state?
Man, that would be such a great study to do. Moran, I see you looking at me. File that one away, right? You're the guy that could
Yeah,
theoretically do that.
typing
That is,
ideas. I'm
yeah.
trying to capture everything so I can put it in my head. Yeah, actually, we were thinking about that.
Yeah.
Well, you start with a village and then you end up with a
Yeah.
door, right?
We're trying to figure out a way to image that, maybe to find something like a PET tracer specifically for 5-HT2A to look at the dynamics throughout the different phases of cancer. Maybe survivors versus actively treated and patient in remission. Yeah, it's a big issue now, like to see where exactly what part of the brain are affected. I know that um, Robin showed some data on the Robin Carrot Harris about the modularity, but to me it was always fascinating the discrepancy between the different you know um, timelines, because the effect when people get out of the experience is almost instantaneous, and the thing that
Mhm.
happens in the brain doesn't happen that fast, those structural changes. So I don't know how to wrap my head around it, but we still have a lot to learn, and it might be immediate effect on the receptor expression and maybe what's downstream to that. I don't know.
Yeah. Yeah, we don't know. I mean, there's a 5-HT2A receptors. There was this old idea that that's how antidepressants work, was that they downregulate the 5-HT2A receptors. They're very much involved in stress responses. And I've got colleagues who think that is a mechanism of action, a rapid downregulation of 5-HT2A receptors. And that may be, if you want to do away with all the sort of psychological stuff, that maybe that is what's, causing the mood effects. People are looking at that. Again, I privy to some things. There's some really interesting work. We'll see, it's interesting.
Well, thinking about sort of the psychoneuroimmunology part, the immunology part, what are the data looking at the potential effects of signaling through the 5-HT2A pathway and effects on inflammation, effects on immune function, immune response, immune infiltration to tumors, for example?
Moran, that's you, buddy. Don't
I
ask me.
think
I'm
we
just
have
a
Greg, actually.
psychiatrist.
If someone knows, it would be Greg. Greg, you're doing some stuff on. Well, I don't think Greg, you're working on psilocybin directly, but you're working on other, tryptamines and psychedelics, and you're looking at the immune markers in the bloodstream, right?
We look at the blood and the brain, and actually, it's very interesting. We use iPSC derived neurons, and actually, what we see is even though it's neurons alone in culture, most of the signals that come from ketamine and from psilocybin and LSD are highly correlated. Like the differential gene expression is very highly correlated, and the vast majority of common signals are actually immune signals, even in a neuronal monoculture. And we see similar core regulators related to like monocyte and NK cell activity, but that's in the brain. And we see some of the common same receptor or cytokines in the periphery. So again, like Dr. Raison was saying, this might be a transient immunomodulation or not big at all, but it's very interesting that in neurons alone, the primary thing we see with all of the rapid acting agents is inflammation related.
What,
Yeah.
what would you what would you expect to see if you were to build, say, neuronal immune organoids? Where you have these together, and you're probably doing this and asking whether or not, uh, even if the neuronal signatures are very similar across the different psychedelic, agonists, the crosstalk with immune partners may differ, or in some way changing the immune functionality to, like, say, a PAM
There's
or a
actually
BAM.
data on this. It just came out last month in Nature. So sorry to interrupt, Chuck, but I don't know
Yeah,
if you
please.
saw this paper published. I think it came from Harvard that they showed that psychedelics control the neuroimmune interactions in fear response, and
Yeah.
yeah, and
Very
they found
interesting.
actually the same things that Chuck was mentioning earlier. IL-6, interferon-1, IL-1 receptor, all the immediate aspects, and like Greg mentioned,
Right.
it's very interesting that those neurons actually express mostly immune-related things in response to 5-HT, to a serotonin,
Yeah.
response, receptor
Yeah.
signaling. So I think that it makes a lot of sense, and it's actually the same thing that we're seeing in nerves peripherally when we're looking at how nerves respond to cancer. Not with no psilocybin or other tryptamines in the system, just cancer nerves. You see that the nerves acquiring those immune transcriptional programs that are heavily enriched in interferon 1 and IL-6 that drives tumor progression. So it's
Hmm.
really interesting to see this association, in the presence or absence of psychedelics, but when those neurons are stressed and how they respond to it.
Hmm. I think it's really interesting. I want to bring this back to Chuck, if I could, for a moment. This idea of homeostasis and returning back to your set point, and whether or not, like you mentioned, IL-6 goes up, it needs to go down. If it stays up, that's not a good thing. I just wonder whether or not some of these psychedelics can assist in in permitting, sort of a return to baseline, if you will. You might argue that depression is sort of like this sort of chronic deviation from a homeostatic control, and that maybe some of these very strong acting ligands can sort of reset the system in a way that allows it to go back to homeostasis.
Yeah. I mean, or go to a place that we would consider to be more beneficial. So a
Yeah.
lot of the work, and this is metaphoric language, not molecular language, but a lot of the work that we've done, and a lot of these more kind of ancient ways of doing things, frankly, like psychedelics or heat, they seem to function as adaptive stressors.
Hmm.
So for instance,
Interesting.
psychedelics. One, we're trying to give people psychedelics while they're sleeping, in deep sleep, to see if they sleep through the experience. Do they still get the benefit? And one of the to try to separate consciousness from biology. One of the challenges has been that these agents cause a massive increase in heart rate, blood pressure. They wake people up. We've had to, it's interesting. They're stressors. There's no doubt about it. And some of my colleagues here at UW have shown in animal models that you can block the sort of short-term anti-anxiety effect of psilocybin if you block the HPA axis response to the drug, right?
Hmm.
So, and of course, heat is, that's a massive stressor for people. So one of the sort of ideas that's floating around, and it really is sort of a hormetic idea. I mean, it's not new to us, although, you'd mentioned Robin Card Harris, and he and I have been talking about this, that some of these type, as opposed to something like an SSRI, some of these sort of rapid acting treatments, it's like they push the organism into the pathology in a way, if I can put it that way. Or you think about psychedelics, they often induce very difficult psychological, stressful experiences, and yet there seems to afterwards be this sort of relieving overshoot that resets. To your point, Monty, about resetting something. We see that very clearly with the hypothermia work, right? That
Hmm.
in fact,
Hmm.
it actually, if we treat people with in our heat machine, a week later they're not hotter; they're actually colder. We've sort of sensitized this system. So
Mhm.
yes, I think that this is really one of the ways of thinking about it. Is these acute treatments, at least some of them, they cause a compensatory overshoot in the sort of opposite direction of their immediate effects, and yeah, whether it's a homeostasis or an allostasis, that it kind of, I mean, Robin Card Harris again has sort of said, that you think about the brain like a ski slope, and if you're, after a while, if you're always following your tracks down, then you kind of get locked into it.
Right.
That something like a psychedelic is like a fresh coat of snow. That would fit with the same idea, right? That you're basically you're causing a reset, um, but that you may be causing a reset by pushing the system into the direction of the problem to a certain degree for a certain amount of time, because you can overshoot that direction, screw things up. But and exercise does that, right? You look at exercise, it breaks things down before it
Mhm.
builds things up, right? That's the sort of
Mhm.
just a classic example of one of these adaptive stressor type, things that has therapeutic benefits.
Yeah, I'd be very curious to see if some of these agents affect, like cycling. Not cycling on a bike, which of course is always good,
Uh,
but cycling the sort of like circadian rhythms. There's just some of these things where there can often be dysregulation, and this to this point of sort of returning to normalcy, if you will, in a physiologic sense,
we have
is
some
somehow
we
mediated by the.
You definitely want
So,
to check out
so,
the New
so
Yorker.
Chuck, Greg, and Moran, this is just super
Yeah, no,
interesting.
I saw like the New Yorker today had like the interview with Mike and Paula, and you mentioned this, Chuck. It's pretty fascinating to see, like how your um, intention settings and how your pre notions about your life and even your beliefs kind of changes the experience, and down the road changes the impact on you. Yeah, well, I'll let you wrap up, Monty. But Chuck, thank you. That was really fascinating. You're going to be one. Yeah,
Absolutely, absolutely. I think just sort of bringing life history into this and experiences and understanding not only pathophysiology but also spirituality and mindfulness and building a physiologic roadmap for that. I think is just a great opportunity. We so much look forward to hearing more about this at the conference
I'll be
in
there.
Texas in
Yeah, I'll be
February.
there,
So
man.
thank you
thank
very
you.
much.
Thank you.
Thank you.